Okay, welcome. So to today's lecture, we're going to be focusing on microbial environments, as well as the communities that are in them, and looking at methods and ways to describe microbial communities. So fun stuff. Okay, let's dive in. So first, we're going to start off with a quick review on energetics, because the reality of understanding microbial environments, understanding microbial communities, is to sort of understand their metabolism. Because one of the kind of fun things about microbes, as we sort of talked about, is not that they're diverse in terms of what they look like, but they are really diverse in that metabolism. And that diversity of metabolism also translates to the diversity of places they can grow. But as we recall, the foundation of all energetics is based upon reduction potentials. So the idea being here is that there's a reduction potential is a measurement of the tendency of a chemical to acquire electrons or to lose electrons and thereby be reduced and or oxidized respectively. Now, the idea that sort of simplify this, what it means is basically some chemicals want to give up their electrons and others want to receive them. And the idea being is if you want to move electrons between atom A and atom B, the, the ultimate reaction to do that gives off some form of energy. And that energy that is given off allows the microorganism to do work, i.e. create a proton gradient. And remember, the proton gradients is what we're really looking for here. We want a proton gradient so we can generate proton motive force. So thus we can generate ATP or adenosine triphosphate. Now, we spent a lot of the past few classes talking about this electron tower. And so we could put the reduction potentials on an order. So we have good electron acceptors here as we go from the top to the bottom, with oxygen being the best, and good electron donors as we start with water here on the bottom and move all the way up to the top to glucose. Now, the idea here, remember, is we're pairing things together. And the greater the differences, so we'd say between oxygen and glucose, which are on completely opposite sides of this tower, increases the amount of energy. So pairing oxygen with glucose is much more energetically favorable than pairing oxygen with manganese, for instance, right? That change or that delta in these E naught values here gives us more energy if we do a bigger jump. But remember, there's all sorts of metabolisms in the world of microbes where we're pairing potentially not ideal electron acceptors with unideal electron donors. It just depends on where the microbes live. But let's talk about a very broad classification of how we talk about microorganisms and how we talk about metabolism. So we're going to start with an electron transport chain, and we're going to be our own one. So we have an enzyme here, NADH dehydrogenase, or NDH1. This is our electron donor. So it donates electrons to the electron transport chain, which is what ETS stands for. Then we have a then we have a final enzyme, a cytochrome, and this can be a wide variety of them depending on the organism. But ultimately what happens is NADH transfers an electron, so it becomes oxidized, reducing NADH dehydrogenase, again by donating that electron, and that NADH becomes NAD plus a hydrogen, a hydrogen ion. Then what happens through a series of reactions, because just transferring this electron from the NADH dehydrogenase all the way down to cytochrome is too big of a jump and not energetically efficient. So what we do is we use a pool of molecules called quinones to transfer this electron. So we're doing this electron transport chain, transferring it first from the NADH to the NADH dehydrogenase, and then we pass it down between this pool of quinones until we end up at the cytochrome. And then finally, we reduce oxygen. So we transfer this final electron to oxygen, making it into water. And as we sort of doing this, we are pumping hydrogen ions across this membrane. So this is what we're doing. We're passing electrons down, and as we're oxidizing and reducing things, it's generating energy. And as it's generating energy, it's pumping hydrogen ions across the membrane. And then the final electron acceptor ends up being oxygen. And so this sort of electron transport chain generates this proton gradient, which we then use to power ATP synthase, which, as you'll remember, is the enzyme that generates ATP, essentially by spinning like a motor. So let's just generalize electron transport system. We have some sort of substrate oxidoreductase, and there's some sort of substrate that donates the initial electron. There's some pool of intermediates to carry the electron through the electron transport chain, all the way towards a terminal oxidoreductase. And then we have some sort of terminal electron acceptor to finally accept that electron as it bounces down through this. So let's talk about some examples of this. So we talked about ammonia oxidizing bacteria in archaea, where they have a substrate and a substrate enzyme. The enzyme is ammonia monooxygenase. Ammonia as a compound donates an electron, being oxidized to nitrite in O2. But again, it passes the electron to this oxidoreductase. There's a series of intermediates. It bounces down to the terminal oxidoreductase, which in this case is a cytochrome. And then the final electron acceptor in this chain is oxygen, reducing oxygen to water. The other pathway we talked about is sulfate reduction, where we have some sort of substrate oxidoreductase, where it's transferring a electron from either NADH or hydrogen gas to this oxidoreductase, passing that electron down till we get to a terminal oxidoreductase, um, which in this case is called sulfite reductase. And we use that to reduce sulfate, SO4, to hydrogen sulfide, H2S. And just as a note, H2S is the smell of rotting eggs. So if you go out on a low tide in Boston Harbor, you'll smell this metabolism happening all the time because of the presence of hydrogen sulfide. Nice, sweet smells. Now, next up is denitrification, where we have some sort of substrate oxidoreductase, and in this case, it's the NADH dehydrogenase, where NADH donates the electron to the oxidoreductase, and it passes it through a series of intermediates. Now, the interesting thing about denitrification, as you'll recall, is the intermediates are a series of enzymes that essentially convert as a stepwise process nitrate, which is NO3, to nitrous oxide, which is a gas. Ultimately, the terminal electron acceptor here is going to be nitrous oxide, and that reduces the nitrous oxide to nitrogen gas. So this is what we have for denitrification. It's kind of a special case. It has this really interesting intermediate pool, which powers the reduction of nitrate all the way to nitrous oxide. And this is what this looks like in real time. So we have that NADH dehydrogenase here, passing the electrons down from a series of enzymes, again, stepwise reducing nitrate to nitrite to nitric oxide, and then finally to nitrous oxide, and then eventually N2 gas. So denitrification is kind of a special little process. So with that out of the way, we're going to jump into today's topic, just because it's a really important thing to think about with respect to how microbes live. Because as you'll sort of recall from these past few um, metabolic pathways we just looked at, is that they sometimes require oxygen and they sometimes don't require oxygen, and they require all sorts of different substrates, whether it's sulfur or nitrogen-based compounds. It just depends. Now, the capacity of any microbe to live in any given environment is depending on its metabolism here. So if the environment doesn't have the metabolites that the microbe needs, it physically can't live there. So with that in mind, we're going to move on to the next part, which is looking at these microbial environments. And the questions we're going to answer are: what environmental factors affect microbes? What are the preferences and limits on where microbes can grow? And what is the terminology used to describe environmental preferences and or requirements? And then finally, what are some adaptations that microbes can use to handle environmental conditions? So it's a pretty cool little lecture, I think. It's a much more fun thing to do in lab, I will note. It's fun to assay microbes and how they grow and all that fun stuff. Now, there's an important distinction here to make, is the environment in our case does not equal the habitat. Because when we think about what an environment is, it refers to the conditions in a given place, which is all the living components, i.e. the biotic components, and all the abiotic components, which are the non-living components. So this is what the environment refers to. But habitat refers to the physical space being inhabited. And this needs to physically provide the necessities for life. This includes water, food, shelter, protection from predators, and potentially access to mates if you're something bigger than a microbe that actually mates. So we're going to start with water. Because water is the most important molecule on the planet. Everything we know about life requires water in some shape or form. You, as a human being, are just one giant bag of water. And just as a final note, whenever we look for uh, or hear about water on another planet, it's so exciting because, again, all life as we know it requires water. And we think, at least in terms of our solar system, all life also requires water. So it's really a really important thing. Now, on average, your average bacteria is about 70% water. And this is true for your bacteria. This is true for us. And it's a process that's required for both cellular metabolism, growth, and all chemistry that life does is aqueous chemistry. And that aqueous chemistry just means chemistry done in water. The interesting thing about water as a molecule is it affects the diffusion of gases. And the really interesting thing is gases typically diffuse 10,000 times slower in water than they do air, which is kind of a striking thing when you think about it. But ultimately, water itself is really easy to find in aquatic environments. Some soil. So we have a film of water here in gray, as we can see here. And what we see is black would be our soil particles. And the white is just like air pockets. So what you're seeing here is that in the presence of the soil particle, the water is physically sticking to the soil particles. Water is actually attracted to soil. Most soil particles carry some sort of charge. And then as you'll notice as we go from A to B to C to D, water starts to disappear and disappear. Now, what you'll notice is that even in D, where the conditions in terms of the amount of water is very, very, very limited, water still exists in very small pockets here and here and even here. So water is there even though it doesn't look like it. And this little bit of water is enough to sustain microbes. So even the driest soils you can think of, like say the American Southwest, water still exists in micro pockets, which in turn can sustain life. So that's water. Temperature is another important factor. So temperature is a pretty broad uh, category from microbes. Most microbes can grow over a relatively sizable um, temperature gradient, but most bacteria and archaea have a large temperature range relative to eukaryotes. And so the coldest bacteria we've ever found is at 90 degrees are locked in ice, but liquid water is ultimately needed for substantial growth. So that's really what we need. It doesn't matter how cold it gets. As long as there's liquid water, the microbes will be there and growing. Now, liquid water does exist all the way down to minus 20 degrees in very, very uh, high salinity areas like these Arctic brine channels, as I mentioned here. So it does exist, and very hot and water can exist in very hot temperatures, greater than 100 degrees centigrade, which is the boiling point of water. And water exists at higher than boiling temperatures because of the very high pressures at the bottom of the ocean. And this sort of brings us to this interesting conundrum. Is the limits on microbial growth temperature, or is it actually the presence of water? And if water could get up to over 200 degrees C or below 100 degrees C, I'm sorry, below minus 100 degrees C, the question we can ask is, should life actually grow? And the question is, we actually don't know the answer to that question right now. But we will likely find out probably in the future. And I wouldn't be surprised if you could find a microbe that could grow at 200 degrees, provided there was liquid water there for it. Now, for our eukaryotes, they don't handle hot temperatures very well. Typically, they don't exist above about 50 degrees or so, but they do pretty well at very cold environments. So these brine channels that I mentioned before, you can find eukaryotes living there. in really, really cold things, and the microbes are physically there. So they're growing. So this leads us to classifying microbes as by their temperature. And this leads us to this term phile. And so phile is just a Greek word for loving. And so we can look at a couple different types of microbes. And so on this y-axis, we have growth rate. So the higher the growth rate, the faster the microbe is growing. And then we have temperature here on the x-axis in degrees centigrade. So we have psychrophiles, which like to grow less than 15 degrees C, mesophiles, which grow between 15 and 40 C, thermophiles, which grow between 45 and 80 degrees C, and hyperthermophiles, which grow greater than 80 degrees C. But what you'll notice between all these is they don't just grow at one specific temperature. They grow over a range of about 30 to 40 degrees, but they do have what's called an optimal temperature, which is going to be the apex of these curves. So here for our mesophiles, here for our thermophiles, here for our hyperthermophiles, and so on and so forth. So microbes have an optimal temperature, but they can grow over a wide range of temperatures. So the adaptations that allow microbes to grow at these different temperatures include changes to their structure of their phospholipid structures, the introduction of haploids, and at really high temperatures, changing from a bilayer to a lipid monolayer in your plasma membrane. But ultimately, one of the things that we see with microbes, and one of the greatest effects we see on temperature on microbe growth, is actually how it affects reaction rates. So this is really interesting uh, not conundrum, relationship here called the Arrhenius equation, where we can look at reaction rates scaling exponentially with temperature. Now, the way this works is we have the reaction rate here on the y, and we have temperature, or the 1 over temperature here on the x-axis at the top, and then we have the log of the rate. Now, what you'll notice is that as we increase the temperature, the reaction rate goes essentially up, which tells us, and because of the log of, because of the log scale here, um, and what we see that the log rate versus 1 over the temperature goes up, and the same thing we see for the rate versus the temperature, it goes up as well. So there is a relationship. So the hotter things get, the faster reaction rates occur. And this is just how chemistry works. And so microbes are susceptible to this as well. Warmer temperatures mean microbes can grow faster. Now, we can actually see this in real life. So we have denitrification rates here in um, some crazy um, uh, measurements that we're not going to even try to explain. And then we have temperature here on the x-axis. And we have, and you can see that as we increase the temperature in both of these graphs here at the bottom and the top, the denitrification rate also increases. So it does work in real life. It's not just sort of some relationship between chemistry. This actually occurs in real life. This is actually denitrification in paddy soils. So fun times. So next up after temperature is going to be pH. And so microbes are really susceptible to pH. This is one of the strongest drivers of whether or not a microbe can grow in an individual environment. Now, depending on where a microbe likes to grow, it can be classified as an acidophile if it likes a pH less than 5, a neutrophile if it likes a pH between 5 and 8, an alkophile with a pH greater than 8. But the interesting thing about microbes and their habitats that they live in is that their habitats can have very, very different pH levels. So, for instance, marine waters, such as the ocean, have a pH of about 8. Lakes are typically a little acidic, between 6 and 7. So there's some places that are mine drainages or potentially geothermal drainage waters that have pH less than 5. Most soils are typically acidic, but there are a few that are alkaline. But ultimately, pH affects the solubility and charge of important nutrients. So what do I mean by that? Well, alkaline waters and soils have lots of charged ions, so cations here, sodium and potassium and calcium. In aerobic environments, iron 3 is soluble when it's acidic, but forms precipitates above pH of 3. So if you're an iron reducing bacteria, you want to live where it's acidic. Excuse me. The next sort of discussion here is going to be about salt. And so salt itself varies wildly in the environment. So we know that water exists naturally that can be potentially distilled, so with nothing dissolved in it basically, to almost near saturated, which for sodium chloride is about 35% salt. And just to put that in perspective, what that means, if you took a gallon of water and you made 35% of its weight salt, that's what 35% saturation means. But the reality is that some microbes really hate salt, and the many of them organisms that hate salt are gram negatives. However, gram positive bacteria like high salt pretty well. However, there are some organisms that actually really like salt. And in fact, there's a group of bacteria called the halobacterium that actually require salt to physically grow. However, we actually have been using salt for years and years and years to cure our meats and fishes to actually prevent microbial growth. So how does this work to kill microbes? How does salt affect microbes? Well, it's not really the salt that affects the microbes, it's the water. And so the idea here, there's this general idea in chemistry that water follows salt. And so if we have a solution here that is isotonic, it means there's equal abundance of large black things inside and small black things inside and outside. But however you see here, um, in this hypertonic solution where we have much more small black things outside the cell than inside the cell. So there's a concentration gradient here, right? So we have an imbalance. So what ends up happening is that the water moves from areas of low concentration to areas of high concentration. And then we see that, and so what ends up happening is that the water ends up rushing inside or outside the cell. And then we have hypertonic solutions here where we have an imbalance, right? We have much more larger black things outside the cell than inside the cell. And water, again, the way water moves is from outside the cell, in this case from inside the cell to outside the cell, because it's moving from a low concentration to a high concentration. And in this graph, it's moving from a low concentration to a high concentration inside the cell. So water moves like thus. And just as a note, the way this ends up working is if you have a microbe that's in an isotonic solution, it does just fine. And as you can see here, this red blood cells are doing just fine. You put it in a hypotonic solution, water rushes in, causing the cells to rupture, which you would imagine would kill the microbe. And then in a hypertonic solution, you see that the cells get dried out, water leaves them, they get all desiccated and sad and shrink. So you can imagine, unless the microbe's in an isotonic solution, or it can make itself in an isotonic solution, it's going to have a bad time. Now, the way microbes handle osmotic concentrations is raising their internal solute concentration. And so there's a number of mechanisms that microbes have, including pumping inorganic solutes, i.e. potassium, or making organic solutes such as glycerol, which is called making of compatible solutes, which don't interfere with cellular function, but balance the osmotic pressure or balance the salt. We have just as much salt on either side. And so this is what we're doing to help balance this. Now, one of the interesting things about this is we can actually see this in real time. So for those of you that ever bought honey, honey almost never goes bad. And so honey has very, very high osmotic pressure. And it's not due to the salt, but it's actually due to the high concentration of sugar. And honey's due to the high concentration of sugar. Now, the interesting thing about that is because of the high osmotic pressure inside honey, it makes honey safe to eat almost indefinitely. So when they find honey that's hundreds of years old, sometimes even thousands of years old, you can still eat honey, and it's all safe because of the high osmotic pressure, which is pretty cool. So next up, we have oxygen. Now, oxygen, as we've talked about, plays a really important role in the metabolism of microorganisms, right? It's a great terminal electron acceptor. It's something that's required for almost all eukaryotic microbes, and it is required for some bacteria and archaea. So we get a little bit of terminology here. We have obligate aerobes, so they require 100% of the time oxygen to be present, i.e., in the case we have Mycobacterium tuberculosis. However, there are many bacteria and archaea that do not require oxygen at all. And in fact, oxygen can be toxic to them. And so we have what we call obligate anaerobes, meaning they always need to be in the absence of oxygen to survive and to live. So example of this would be Clostridium tetani, so the bacteria that causes tetanus. Um, in addition, we also have microorganisms that are facultative. So E. coli, the microorganisms we talked about quite a bit when we talked about gene regulation, uses oxygen if it's around, but it's okay if oxygen isn't around. And there's actually sort of this interesting, sort of crosstalk where they can switch between both of them. And in under certain conditions, they grow better. Other conditions, they grow better with or without oxygen. So it's interesting. And then finally, we have microbes that like a little bit of oxygen, but not a lot of oxygen. And these are what we call microaerophiles. And so, for instance, Streptococcus, the bacteria that causes strep throat, or I'm sorry, I should say the genus of bacteria that causes strep throat, they like around 4% oxygen at best. Um, that's about the oxygen that's present in the back of your throat. So they like that. They like that environment there. Now, what we're looking at here is some more terminology where we have a few key terms where we have oxic, which is greater than 30% saturation. And this is relative to the saturation of oxygen in the atmosphere. So there's a consistent amount of oxygen in the atmosphere. And when we say saturation, it's all relative to how it compares to the saturation of oxygen in the atmosphere itself. So that's oxic, above or equal to 30% saturation, hypoxic, which is 1 to 30% saturation, and anoxic, which is less than 1% saturation. Now, how much oxygen there is in an environment is affected by the diffusion of oxygen through liquids, mixing, and the activity of oxygen producing photosynthetic organisms. But in generally speaking, as you go deeper, whether it's in soil, whether it's in the water, oxygen decreases with depth. So we can see this graph here at the top where we have depth goes from 0 at the top all the way down to 1,500 meters. And you can see all these things change, but you see oxygen dips dramatically, basically goes anoxic, and then it comes up a little bit. So again, oxygen generally decreases as you go deeper and deeper. Now, in soils, oxygen penetrates through the space between soil particles and doesn't really penetrate very well when soil is waterlogged, just as a note. And it's interesting, you can look at really, really small depths. So in this past figure, I showed you 1,500 meters. That's pretty deep, right? But you can actually look at oxygen concentrations in soils that have very, very shallow depths. So in this case, we have 0 millimeters here at the top and 45 millimeters at the bottom. So we're talking about here just under 4 centimeters, so 2 inches. Very, very small. What you'll notice here is that oxygen dips pretty rapidly early on, early on, and early on. And the interesting thing here is you can also see the effects of not just depth of soil, but you can also see the effects of vegetation on the penetration of oxygen into soils. So it's actually kind of cool. You can see this on very, very small scales. It doesn't have to be huge scales like we're seeing here. In soils, it can change on microscopic scales, which I think is really, really fascinating. Our next environmental condition is going to be pressure. So high pressure environments present actually the largest biome on the planet. And where does that biome actually exist? It's actually the deep ocean, which is a huge part of our planet, as well as the deep subsurface of the crust, so the soil you're standing on. Some microbes love, love, love high pressure environments. So microbes that love high pressure environments are called pisophiles, and they will only grow at high pressures. Now, hyperpisophiles grow at very high pressures, greater than 60 megapascals. And so to put that in perspective, the Mariana Trench, which is the deepest trench in the world, it's 7 miles down, if I'm not mistaken. It has a pressure of water above it of 110 megapascals. So really, really pressure. Now, pisophiles are actually kind of an interesting group. They likely evolved from low pressure psychrophiles. And just as a reminder, psychrophiles are cold loving bacteria because to survive under high pressures, you actually need, there's actually a very similar amount of adaptation that you would need to grow at very cold environments because you need lots of unsaturated membrane lipids and similar DNA and protein modifications. So just as a fun fact, being cold might actually mean you can grow at very high temperatures as well. And so those are all the different types. It's certainly not an exhaustive list. An exhaustive list would take hours and hours and hours to go through, but we're not going to go through all of them. There are many, many more. This include light, carbon and nutrient availability, and some we don't even think about or will consider in the context of this classroom. But that's our environments. Our microbes are, as you can tell, they grow pretty much anywhere. They will use pretty much anything in their metabolism. So they are really quite remarkable in that context. So the next topic we're going to discuss is how being small matters to microorganisms. Because we talked about microbes are small, right? That's in the name. But how does being small matter? And the way being small matters for microbes is the patchiness of a habitat. And so the question we can ask, what is a homogeneous environment? And the answer to that is an environment that is the same throughout, right? No matter where you go, you have the exact same set of environmental conditions. The question you should ask yourself next is, does a homogeneous environment exist in nature? The answer to this is two-part. The first is for macroscopic organisms like us, the answer is sometimes yes. So for instance, the ocean is a pretty consistent for large patches of the ocean. But at the microscopic scale, habitats are remarkably patchy, which leads to the formation of lots and lots of what we call microhabitats. So habitats that change on very, very small scales. And this is true not just in soil, which are very, very different at the microscopic scale, but even in aquatic systems. So for instance, in water, there's all sorts of particles floating around, which are barely noticeable at the macroscopic scale, but they represent an entirely different habitat and conditions and environment for these microbes to grow in. And this idea of patchiness, even at the small scale and the large scale with many, 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 many environments, helps support the large amount of microbial diversity in many, many seamlessly homogeneous ecosystems. So we're going to revisit this idea, but I just want to show you what I mean by this um, about patchiness. And so we have a bunch of soil here, and you'll notice the scale here is 10 microns, so not a very huge, sample size we're looking at, a few hundred microns across. But what you'll notice is that there's lots and lots of variability at the small level, right? You can imagine this little spot right here is very different from what's over here, right? And this is what the patchiness we're talking about. At the microscopic scale, at which bacteria would see this as huge, you can have very different conditions here versus here versus here versus here, right? And that's all on 50 or so micron scale. And remember, for bacteria, that's huge, right? 50 microns is big for a bacteria. Very, very small for us, but big for a bacteria. So this patchiness leads to not only lots of environments, but leads to a huge amount of microbial diversity as well. Now, microbes themselves are very, very small, which is obvious from their name. Now, cell size, as we've discussed, affects the transport of limiting nutrients. It also affects predator-prey interactions, macromolecular composition, and many other aspects of microbial biology, as well as their ecology. And navigating the world is different for microbes too. And so one of the ways microbes experience this, and it's kind of a weird thing to think about, but how microbes navigate water is very, very different for us than it is for us. And when I jump into a pool or when you jump into the pool or the ocean, we could just swim right through it, right? But if you toss a microbe into water, water is very viscous to them. And so the size of the microbes, because they're small, affects their capacity to move through water. And so this leads us to this idea of a Reynolds number. And a Reynolds number is the function of size, velocity, density, as well as viscosity. And it's basically a ratio of the internal forces, so that forces of the microbe versus the viscous forces. And so let me just tell you what this means in the most simple terms I can. Big organisms like us have a very large Reynolds number, which means what affects our movement or what limits our movement is our internal forces, right? Our own limitations, our own bodily limitations. Now, small microbes or small organisms have a low Reynolds number, which means viscous forces, not their own internal forces, win out. So just to put this in perspective, is that diffusion is also much more difficult at the small scale. So if you're small, molecules can diffuse quickly through your whole cell. And so we can just look at this little handy table. We have the distance, 1 micron, 10 microns, so on and so forth, and the diffusion of oxygen and glucose. And you can see as we increase the distance, um, We have this is through an aqueous solution. I should mention uh, oxygen and glucose. It's easy at small scales, right? Our microbes at maxing out at about 10 microns, glucose can diffuse relatively rapidly through them, which is good if you're a microbe, right? If you need glucose or you need oxygen, it will diffuse through your entire cell almost instantaneously. However, as you notice, as we go up to 1 meter, for oxygen or glucose to diffuse for 1 meter, it takes a year or 3.3 years. So being small is really, really good because diffusion means things are more likely to move into your cell, means you're more likely to be able to uptake things quicker and thus respond quicker to the environment, which is a really cool thing for microbes, right? They're small, the environment's tough for them, but they have this really rapid diffusion. And size and being able to pick up something like oxygen or glucose, which might be limiting, is actually really important for microbes to live in very, very nutrient poor ecosystems. So, with that out of the way, we're going to just sort of summarize this previous section that we went through. So the big key takeaways here are microbes are affected by many environmental conditions, including temperature, pH, osmolarity, pressure, oxygen, et cetera, et cetera. Any condition you could think of, it affects microbes in some way, shape, or form. Remember, when you're that small, pretty much everything affects you. Every microbe has a range of its optimum growth, and we can classify organisms by this, right? We can classify as a microorganism as a hyperthermophile if it likes really high temperatures. But for microbes to live in any number of environments, they have a series of adaptations to deal with these conditions that include changing membrane fluidity to modifying their internal solute concentrations. And while being small makes things difficult for microbes, it does give them some advantages for the diffusion and the uptake of nutrients from the environment. So it's kind of a double-edged sword being small with microbes. It's either good or it's bad. It just depends on who you're asking. So with that out of the way, let's move on to the next part of this lecture, which will be talking about methods and how do we study microscopic organisms. Now, if we were in the laboratory or as you've been progressing through your laboratory exercises, it's all there's a number of ways to study microbes, and the most common way we do this in the laboratory setting is growing them on there. But science as a process is all about collecting data. And there's lots of different types of data microbiologists collect daily, and these include what metabolites the microbes are producing, their interactions, some regulatory pathways, and etc. But for the purposes of what we're going to talk about, we're going to focus on two. So we're going to focus on community composition, so who's there at any given time, and abundance, how many are there. And to do this, we're going to talk about the two distinct types of microbiology out there: cultivation dependent, which requires the direct growing of microscopic organisms on a physical media, i.e. a petri dish, or cultivation independent, which doesn't require any culturing, which is prominently featured by looking at DNA sequences. So we're going to start with culture dependent, and it requires growing microbes on a petri dish. So when you've been looking in the lab, you've been growing microbes on petri dishes, right? Or you've been visualizing the growth of microbes on petri dishes. This is an old school method, right? This is how we first started studying microscopic organisms after we started looking at them under the microscope. Even though it's old, it's still very, very, very useful. It's a very useful technique because once you have a microbe on a plate, like all these microbes here, it allows you to manipulate the growth of these microbes to figure out how much oxygen they like, what temperature they like, what nutrients they like, what types of carbon sources do they like. So it's a really important thing to be able to grow microbes, but we are limited by our capacity to grow microbes. So current estimates is that less than 1% of all known microscopic organisms out there can be cultivated. So that means that there's 99% of all species of microbes out there that we physically can't grow on this plate. And we'll discuss how we study those unculturable microbes in the latter part of this section. Now, culturing of microbes is great because remember, once you have it in a culture, you can start messing with it. You can understand all of its biology. And culturing is our best and only truly reliable method to gain information about what microorganisms can do. And it's very useful even still for identifying microscopic organisms. So if you go to the doctors to be tested for strep throat, they identify that you have strep throat by growing it on a petri dish. They don't look at its DNA, they grow it on a petri dish. So it's still incredibly useful, not just for any old garden variety microbiologist, but it's also really important in pathology, and medicine still as well. It's still a really important thing. I don't want you to, this first part of this lecture to get lost in all the cool technology of the back end of this lecture, but it is still a really important thing. And so one of the more important things we're going to think about is what we call the plate count method. And this is, we can grow a microbe on a plate and identify it with metabolic tests, but we want to know how, if we can use culturing to get an idea of microbial abundance. And the way we do this is by the plate count method. Um, but the sort of interesting thing here is that it does severely underestimate the number of microbes present. Because the way the plate count method is, it makes the assumption that one colony of a microscopic organism that you see on a petri dish is a result of one bacteria landing on that specific spot and growing exponentially to produce that colony. So that's one way we underestimate it, because sometimes there's multiple organisms that land and make a single colony. However, we also know that we can only grow a small fraction of what's present in any given microbial community, right? Because we know that 99% of bacteria out there are not culturable. So it's sort of this give and take, right? It's good for counting, but it is overall limited. So the way this works is we take some original culture of bacteria, and this could be like a culture you're growing in the lab, or maybe you take up like a gram of soil and you mash it up to get all the microbes off of it, and then you do what's called a serial dilution. So you take 1 milliliter of the original culture, you do a 1 to 10 dilution in fresh broth, you mix that up, and then you do another 1 to 10 dilution. So you transfer some of this to a brand new tube um, with 9 milliliters of broth, you mix that up, and you keep repeating this over and over again, doing a dilution. So you do a tenfold dilution here, a tenfold dilution of that, and so on and so forth, until you get all the way down to a 1 over 100,000 fold dilution. And then what you do from there is you plate a small fraction of that, in this case, 0.1 milliliters or 100 microliters onto petri dish, and then you count how many colonies. And remember, the idea here is that one colony, as you can see here, is the idea that one microbe landed there and grew into the big piece of bacterial colony that you see here. So that's what we do. And the idea being is we can count how many colonies we have and multiply it by this dilution factor. So in this case, I have 6 colonies. I have a tenfold dilution here by only plating 0.1 milliliter. And so what I end up getting is zero point 0.1 milliliters, which is tenfold dilution, plus 10,000 fold dilution, making me have 100,000 fold dilution. So I take that dilution factor and I multiply it by how many colonies. So in this case, I have 6 times 10, um, 10 to the 10 to the 5 microbes per milliliter of culture. So again, I can use this dilution factor, count how many colonies I have, and multiply the amount of colonies I have by this dilution factor. So it's a really handy way to get a pretty good estimation of how many microbes you have. And it doesn't particularly work great for complex environments like soils, but counting how many microbes you have in your own colony, in your own culture tube in the lab, this works fantastic. So it is pretty good. Culture dependent method. So we answer these main questions. Who's there? By cultivating the microbes. And then we use a series of tests and we compare it to known microbes to get an ID of your isolate. In this case, we can compare it to this really handy manual called Bergey's manual. And then we can also count how many. And so we use the plate count method itself to calculate how many of each isolate are present in our original sample. So we can answer both of those questions, who's there and how many. And just as a note, as we've discussed, they are limited, but they still are effective at understanding how many microbes you physically have there. So it is overall useful. Now, what we're going to be spending the rest of the lecture on is looking at culture-independent technologies. And these are methods that don't rely on cultivating microbes, right? We don't have to grow on a microbe on a plate to understand its biology. And we do this for these culture-independent, or what we would sometimes call molecular methods. Now, the backbone of culture-independent technology is based upon the study of nucleic acids, principally DNA and RNA, but we can also use lipids, fatty acids, as well as proteins and metabolites to also understand microbes in the environment without physically culturing them. With the workflow basically being take a sample, extract the DNA, extract the RNA, extract the proteins, extract the metabolites, extract the fatty acids, and analyze them. So that's the gist of it. But if we're studying DNA, we need to actually oftentimes we don't just study all the DNA present. We typically study one specific region of the DNA. And to study one particular region of a bacteria's DNA or its genome, we have to make multiple copies of it. And we do this by a technique called PCR, or polymerase chain reaction. Now, molecular methods themselves rely on making tons and tons of copies of a specific region of a DNA. We need to make enough material for analysis, so we need trillions of copies. This method of making trillions of copies is, again, PCR, or polymerase chain reaction. Now, PCR is an interesting technique because it uses the same machinery cells use to replicate DNA: primers, DNA polymerases, buffers, things like that. PCR uses all of them. Now, for primers are different than, you think about primers used to replicate a bacterial's own genome, but primers tell the polymerase where to bind to the DNA. It's very much the same way they do in replication. However, primers used in PCR actually target a specific region or gene. So in the case of what we're talking about here is the 16S ribosomal RNA gene, you can design DNA-based primers to target and amplify the 16S gene to make billions of copies. And I'm sorry, I should say trillions of copies, because we really are in the trillions here. So that's what's happening with PCR. We're making trillions of copies of a specific region. And so there's, 16S is the sort of the standard, but you can also use PCR to understand biomarkers. So looking at genes such as NERS, which is a biomarker for denitrification, or DSRB, which is a biomarker for sulfate reduction. So it just depends on what you want to do. You can design PCR primers to detect any gene in a bacterial or even a eukaryotic genome that you wanted to. And so there's a special technique called quantitative PCR, or what's sometimes called real-time PCR, that we can use to quantify the amount of DNA or the amount of a gene product in a given sample. Now, I'm not going to dive too deep into this, but just know that anytime you see qPCR, or quantitative PCR, what you're doing is calculating the abundance of your PCR product. So, you could think it's like 16S. In a given sample. So you extract the DNA, you perform quantitative PCR, and it tells you how many copies of a given gene you have in a given sample. So it gives you a relative idea about abundance, and it answers this question of how many microbes are in a given environment. So those are ways we can study specific genes, we can get the abundance of microbes, but once we get all these PCR amplicons, so in this case, we have all these 16S ribosomal RNA genes, how can we answer the question, who's there? Well, the methods we do this are basically just sequencing technology. There are some older methods, you might see these as you're reading about your microbes, including DGGE, or denaturing grading gel electrophoresis, or microarrays. Those are old ways, but as I mentioned, the most common ways to understand which microbes are present in an individual sample is by DNA sequencing. Now, as we've sort of progressed through time, sequencing has gotten better and better and better, and technology itself as a whole has gotten better and better and better. Now, DNA sequencing was invented in the late 70s, and so back then we used to generate one strand of DNA at a time, but now we generate millions, tens of thousands at a given time. So DNA technology has progressed really, really dramatically. Now, when we're talking about DNA sequencing in respect to microscopic organisms, we're typically sequencing that 16S ribosomal subunit gene. And this gives us the taxonomic identity of the microbe that you're looking at. Now, the idea being the 16S ribosomal RNA gene is the Cadillac of biomarkers. And the reason for this is that all organisms have ribosomes. So every organism on the planet has ribosomal RNAs. And the interesting thing about 16S is because every organism has it, it does have a high degree of variability. So there's regions that are really, really variable between different taxonomic lineages of bacteria. So as you go from, say, gram-positive to gram-negative bacteria, 16S varies between those two large groups. And that's useful for identifying them taxonomically based upon their DNA sequence. But the other thing about 16S has some pretty heavily conserved regions, which are really good because they're remarkably similar across a wide variety of taxa, which is good because if you're trying to PCR amplify them, you want to be able to have a really hot piece of your DNA to be very, very similar across many organisms. So you only have to design one PCR primer to target multiple groups of microscopic organisms. And just as a note, 16S is how we study bacteria and archaea. If you're going to study fungi, we use a region of the fungal chromosome called the internal transcribed spacer region, just as a note. Um, so in terms of sequencing, there's a couple methods we can sequence DNAs. So the first is what we call a clone library. Basically, it's the oldest method. It generates tens to hundreds of sequences per sample, so not a lot of DNA sequences, though that might seem like a lot of DNA sequences if you've never played with DNA sequences. But trust me, as someone who's played with a lot of DNA sequences, 10 to 100 is not a lot. Next up is 454 pyrosequencing. And this is the very first generation of what we call high throughput or next generation sequencing technologies, which could take a pool of 16S genes and essentially generate thousands of sequences per sample. So that's pretty handy. However, modern sequencing, which we would call next generation sequencing or the current methodology, can take 10,000 to 100,000 16S sequences per sample. And if you just sequence one sample on a sequencing run, you could actually get millions from that single sample. So really, really good sort of uh, easy, sort of a wide variety of technologies, but the most common we see nowadays is this next generation sequencing. And so Sanger sequencing was the original. So if you want to read up a little bit about this, I'll leave this slide here for you. You can see how the sequencing technology of Sanger sequencing works. Really, really sort of cool method. Really, worthwhile if any of the stuff I've been saying is interesting. Now, sequencing itself, the methods themselves change rapidly. So, when I started grad school in the early, the early 2010s, we were using one type of sequencing. And by the time I graduated from grad school, we were using a completely other type of sequencing technology. So it just varies. Now, the basic process of analyzing your data, so turning a DNA sequence or a bunch of DNA sequences into data, first requires you to clean up data. Now, the way we do this with DNA sequences is basically we get rid of low quality DNA sequences in what we call chimeric sequences. Now, chimeric sequences is a DNA sequencing mistake. It's basically two distinct sequences, i.e., like maybe one from E. coli and one from Bacillus merged together into one sequence. That's all a chimera is. But that's the first step, getting rid of poor quality and bad data, because your data analysis is only as good as the data you put into it. Once you do that, you condense the data. You have to call a species level definition. And the way we do this for microbes, and the way we've discussed slightly in this class, is picking what we call operational taxonomic units, or OTUs. Now, the way this works is you cluster them based upon sequence similarity. And the sequence similarity is typically 97% sequence identity. And what this means is if I have two DNA sequences, both 100 bases in length, if they have only two base pairs that differ between sequence A and sequence B, based upon my OTU definition of 97%, these two sequences would be 98% similar and would thus be considered the same OTU. Now, the same comparison, two DNA sequences, both 100 bases in length. If they have four DNA bases that vary between them, they're 96% similar, and thus they would be considered different OTUs. So it's basically a way for us to say this is species A and this is species B, strictly dependent on the DNA sequences there. And to do this, there's lots of computational methods out there. There are many, many algebra-based algorithms out there, and it's very, very common. There's also some brand new technology out there called amplicon sequence variants. We're not going to discuss, but just to note, it is out there. So once you get your OTUs, you have to assign taxonomy. So you compare your OTUs to a database of known sequences. So you say, I have sequence A or OTU A. I compared its similarity of its DNA sequence to a database of known microbes. It was of high similarity, in this case, to Paracoccus denitrificans. That's what we do. So we give each OTU a taxonomic identification as close as we physically can. And then finally, we make a gigantic OTU table. where we have OTUs. So you have OTU 1, 2, 3, 4, 5, 6, 7, and their taxonomic identification here. Those are our rows. And then we have the columns are different sites. So site 1 could be like my armpit, site 2 could be my elbow, site 3 could be the bottom of my foot. That's what we mean by site. And what you have here is the numbers are the abundance of the microbes was found in that sample. So for instance, for site 1, I have 92 of OTU1, 370 of OTU2, 51 of OTU3, and so on and so forth. So we get this big matrix. How many times was each OTU, and we know its taxonomic identification, found in each one of these sites. So it's a really handy sort of table to use. Now, once we have this table, we could use the OTU table to calculate a couple different things. So the first we can do is calculate what we call alpha diversity, which is the diversity within a given sample, i.e., how many different species of microbes do I have? So we have this sort of this very basic example here where it has the number of beetle species in the Thames River. So the y-axis of this graph is the number of species collected and the number of individuals collected, and i.e. the number of beetles collected. And we can see that as we increase the number of beetles collected, we don't find any more species over time, but we do find a lot of species of beetles. And this is what species richness is. How many different species, or in our case for microbes, how many different OTUs do we have? However, we can use our microbes to estimate how many species potentially we have. So there's lots of formulas out there, mathematical in nature, that allow us to estimate based upon how many species we have, the total number of species in a given sample. So we could take, if I have 15, I could potentially estimate that I have 100 or something like that. So we can estimate or calculate absolute diversity metrics as well. So so the sort of the more important comparison here is calculating what we call beta diversity, which is the diversity among samples. So we start to compare site 1 to site 2 and site 1 to site 3 and so on and so forth. So what we do is first by calculating a similarity um, between different samples. Now, the idea being here is what we're looking at to say site 1 is 55% similar to site 2 or 44% similar to site 3 is we calculate a couple different things. So the first is we look at the presence and absence of species. So if I look at between two samples here, what I can do is say, okay, if site 1 has these 5 species and site 2 has the same 5 species, well, that presence and absence data, they would be 100% similar. So that's what presence and absence means, just the sheer presence of microbes in the samples. Now, we can also incorporate this idea of abundance. So if we go back to our table here, I think this table is a really handy one. So when we're looking at presence and absence, again, it's looking at is OTU1 present across all these sites and so on and so forth, right? Now, changes in abundance, what we can do is look at, okay, I have site 1. It has 92 presences of OTU1. Now, site 3 has 370. Site 2 has 376, sorry, and site 3 has 348. So what we can do is compare how the abundances of microbes change and the presence and absence of them to calculate what we call a similarity score. Now, the higher the similarity score, the more similar they are. So in this table here, comparing site 3 to site 2, we see that they are 78% similar. But if we compare site 2 to site 4, we see that they're only 17% similar. And again, to calculate that, it's all about the presence and absence and how the abundance physically change. Now, there's also this other really cool technique, which is called the unifrac or the unique fraction method, which actually takes into account both presence and absence of microbes, but also looking at the phylogenetic similarity of microbes. So the idea being is that the more similar microbes are, the more likely they are to occupy a similar set of environmental conditions or have a similar set of metabolic capabilities, and thus UNIFRAC takes that into account. So it's a pretty cool method, pretty widely used across the field of microbial ecology as a whole. But this is beta diversity, calculating the difference in the abundance and the presence absence of microbes between all your different sites. And then we can visualize this by looking at a couple different graphs here. So we have first what we call the principal coordinates analysis, and then we also have over here the same graph. Now the idea being here is we're going to just look at this very simple graph over here. It's a simple XY depiction. Now, the idea being here is that each one of these corresponds to a distinct sample, with the idea being that, we have sample one, sample two, three, and four, right? The same four we've been talking about. The idea being that the closer they are in 2D space, so sample four and sample one are very close to one another in 3D space. Thus, they have more similar microbial communities. And just to go back, site one and site four, 32% similarity, pretty close to one another. So we can use these, again, these 2D distances to infer similarity of microbial communities. And so this is how we visualize um, beta diversity, just by nice 2D plot looking at similarity based upon how close they are. So these two samples are much more similar than, say, site 4 and site 2. So it's a pretty handy thing. Now, that's how we analyze sequencing data, cluster to know to use, and calculate diversity, and assign taxonomy. It's pretty straightforward and to understand. Now, there are some issues associated with DNA sequencing that we should discuss. So first, there are issues with PCR primer bias. So there's many issues with designing PCR primers, and some of them just are really bad at capturing certain groups of microbes, and thus they underestimate the presence of certain microbes. So just as a note, not all PCR-based reactions are perfect. In addition, PCR, essentially being PCR, essentially requiring PCR is in itself a bias. And because PCR is only as good as the primers you design. And the problem is you only see in the environment what your primers can detect. So it does bias you against them. And the other thing, what we get from our sequencing is not absolute abundance, right? We're not growing microbes on a plate. We're only getting the amount of sequences they have in a given sample. So it doesn't give us total abundance, but it does give us relative abundance. So that is another issue as well. And then the other thing to mention is that it is hard to link the function of 16, the taxonomic information we get from 16x data to functions. So it's kind of a difficult thing. It's kind of a limitation of DNA sequencing based upon PCR. Now, just as a reminder, sequencing, and just to overview this, sequencing gives us community composition. So it answers the question, who's there? And we can use qPCR to tell us about how much, right? And sequencing, again, doesn't give us absolute abundance. It does give us relative abundance, but qPCR plus DNA sequencing is a really good combination to answer the questions of both who's there and how abundant they are. So moving on, we're going to talk about some non-PCR based methods first, and these require us to first talk about omics. And so these are methods that look at all of the things present in a sample. They don't require PCR amplification at all. And so they're not subjected to all those biases that I just mentioned that are associated with PCR. The first one is metagenomics, where we sequence all the DNA in an extracted sample and use the sequences to reconstruct genomes and metabolic pathways in a given environmental sample. This is a really good method to understand how microbial communities work, what their metabolic potentials are, again, without all those biases of PCR. But the reality of this method is it does require lots of sequences. So you need, for a good metagenomic sequencing, you need at least 1 million DNA sequences, preferably tens of millions. So it is much more data intensive. And as you can imagine, it's much more computationally limiting, and it's also much more expensive to do. However, it does give us the benefit of who's there and what functions they're potentially doing. So, which is a pretty good. Next up is transcriptomics as well as proteomics. So instead of looking at DNA like we did in metagenomics, we can look at messenger RNA or the transcriptome, or protein, which we call the proteome or the proteomics in a given sample, which is good because it gives us a snapshot of the metabolic processes that are happening in a given microbial community. So it provides us the answer to the questions about the function of a community, but but at the end of the day, it's still a snapshot of the actual function of the community. As of the recording of this lecture in 2020, it's becoming much more common, but it is still particularly very costly to do a really good analysis of the transcriptome and the proteome of most microbial communities at this given time. I would imagine within the next 5 years or so, it'll become relatively routine as sequencing costs come down. And so that's all of our DNA sequencing based technologies. We're going to move on and wrap up by talking about some pretty pictures. The first one we're going to talk about is actually fluorescent in situ hybridization, or FISH. So it's pretty pictures plus awesome name. Now, this is a microscope-based technique, and what you do is you fluorescently label DNA probes that bind to specific DNA regions in a cell. So what you can do is generate taxonomic-specific probes that will bind to, say, only E. coli or only Mycobacterium or something like that. So it allows you to look at specific microbes in their actual habitat, even if that microbe has been frozen or fixed to a slide. So it's a really handy way. And what fish can do is can give us interesting patterns in the microbial community as they're arranged physically in 3D space. But they can also answer the question of how many microbes are present. And where fish becomes really cool, it's not sort of in pictures like this where we have just one microbe and how it's 3D oriented to other microbes. So pink versus bluish purple here. But when we actually start to combine multiple probes. Now, this is my favorite paper in all of microbiology because this is a study looking at the microbiology of the oral cavity of humans, in particular the dental plaque. And we can see here we have a bunch of different probes. So for instance, green is the bacterial genus Streptococcus, pink is Corneobacterium, and so on and so forth. So we're using multiple fish probes to understand how these microbes are oriented in 3D space. And the awesome thing about this is look at the scale bar here, 10 microns. So we're seeing microbes change on hyper small scales with fish. And this paper is really fascinating to me because of how not just randomly arrayed these microbes are, because they're not. There's a lot of orientations how they are, but just how crazy they change with one another and how they change based upon physical distance as well as the environmental conditions inside your mouth, which I think is absolutely astounding. So let's wrap up this final part. So microbiologists use many tools to answer the question of who's there and how many. They include culture-dependent technologies, which require the physical growing of microbes on the plate to identify who and how many there are. And it is the best way for understanding what a microbe can do, what any given microbe can do, but it is limited by our capacity to culture microbes. Remember, only 1% of all known microbes can be cultured at this given time. Our culture independent technology doesn't require cultivation, and it's mostly based upon DNA sequencing and PCR. But going forth, omics technologies, proteomics, metabolomics, transcriptomics, metagenomics, or ultimately will replace PCR-based methods as they become cheaper and as computation also becomes cheaper and easier to do. So with that, that will be the end of tonight's lecture. And so if you have any questions, please do let me know. But otherwise, I hope you guys have a great time and take care. Bye-bye.